Okay, I think it's close enough to 2 o'clock, I think. Did I say we're starting at 2? Yeah. Okay, so I'm going to take you through a couple of things really quickly that as you hopefully have reviewed over the past couple of days, that if you haven't, you should make sure that you go back over and understand some of the more important things. Just a couple of really quick notes. So I took everything based on the lecture notes that you have supplied for you as a PDF file. You have nothing to worry about from the textbook itself. The majority of the questions that you'll see on the midterm tomorrow are the application types, at least from my section. And I had submitted 14 questions in total. I don't know how many are actually going to be used. How many questions were you told you were going to get on the midterm? 26. Okay, I'm not really sure what that means. I gave 14. I'm not sure if that means that 12 or 13 are going to be used or whatever number of questions is going to be used, or if Dr. Key only has 12. Did he give you more of a breakdown of how many questions? No. So I had 7 from each of the lectures. Most of the questions that I have, again, come directly from the lecture notes themselves. They're application or integrative type. So which of the following might not be found in the intermediate zone? That would be sort of an integrative type of question, right? So you need to understand the spinal cord, and it's not about memorizing one slide. It's about knowing what's in the intermediate zone of the spinal cord, what's in the dorsal horn, what's in the ventral horn, and then being able to apply that knowledge across multiple slides. So again, it's pretty evenly split from my perspective between the first 3 lectures I gave you, first 3 lectures in that first lecture I gave you, and then the second lecture of 3 different hours in the last section was also about 7 questions, and you should know some of the following details. Do you have any questions about the format of the multiple choice that I submitted before we go on? No? Looking forward to it? Yeah? Do you have a question?
There's no case questions, like for example, let's say a patient has
No, that's an application type of question. So are those types of questions ones that will be asked? Yes. Yep. Yep. I call them application. If you want to call them clinical case studies or whatever, yes, they're more of that type than any other type. Any other questions? No? So you will have questions, and you should expect to have questions related to the CNS as well as some of the basic neuroanatomy. You should know all of the different cell types that we've talked about, some of the functions, especially with the production of different CNS molecules, and you should make sure that you know what different supporting cells have in terms of their function, etc. Okay, so if that's completely alien to you, you should make sure you go back and look at that. Should also look at labeling methods to visualize the different compartments. Do you know what I mean by that? No? So today is a good time to ask questions. I had mentioned, for example, that you could identify different glial cells that are found in the CNS versus those that are found in the PNS. That slide should sort of ring a bell with you. There are some marker proteins that you can label that are more specific to the CNS versus the PNS, and that's what I mean by labeling. You should be able to identify what proteins, what types of things are found in the CNS versus PNS, and what are the associated cell types. And again, you should make sure that you're familiar with that concept. Related also to the first series of lectures, the first 3 lectures, you should also know some of the basic properties of the neuroanatomy that we went through as well. I had mentioned in one of the slides that you had 6 different funiculi. Again, it's paired on both sides. You have one funiculus here, or tract, same idea, same word. We call these dorsal columns medial lemniscus. Over here on the side, we call it the lateral funiculus. And then we also have this ventral funiculus. That makes up 1/2 of the 6. And then it's repeated over here on the left. And that's how we come up with the 6 funiculi, or the 6 different regions of the white matter that surround the central gray. And again, you should be familiar with the overall patterning of the different regions, what the different regions are sort of responsible for. So the intermediate zone, what would you characterize it as being responsible for? What does it do? So basic processing, so some integration, some processing of information. So where is that information coming from into the spinal cord? So in the dorsal horn, in the dorsal gray, it might send projections down into the intermediate zone. Again, just to give me an idea of how much review you guys have already done, what kinds of cells might I expect to find in the um, intermediate zone? Any ideas? Like this was one of the slides that we covered at the very end of this particular section. So at the very end, yes. No, it's not just myelinated axons. We would expect to see, for example, myelinated and unmyelinated axons out here. In this region, we would expect to see unmyelinated axons as well as short cell processes and some axons, but not generally speaking, this is the central gray region, and we don't have very many myelinated axons. We might expect to see other cell types like the wide dynamic range neurons that are found here. Remember, that's a processing type of interneuron that we talked about at the end of the last sort of lecture slides related to somatosensory processing. Does that ring a bell for all of you? Wide dynamic range neurons, WDRs, they would be found in this sort of intermediate zone region. Any other questions before I go on, or anything that you want to go over in this particular series of slides related to the spinal cord? White matter, gray matter, processing. So this is a slide that you should probably spend some time reviewing a little bit. So there are different tracks that we can find in different regions of the spinal cord, wherever we're taking a look. So we send information going up, and we talked about specific tracks in terms of how they send their information up the spinal track, up the spinal cord, and we had talked about that specifically in the spinothalamic. So it goes from the spine up to the thalamic region of the brain, and that's involved in sending information up through the spinal cord. And you should also know some details there, and I'll go through that in a moment. What other ascending tract do you remember? So sending information up along the spinal cord up into the brain from the periphery. Should I be worried about tomorrow? A little bit? No? Hopefully not too much. So we also talked about two other systems in that lecture on somatosensory processing. We talked about the spinothalamic, as I mentioned. There are specific cells associated with those. You should go back and review them. This is a good slide for descending information, but I just wanted to, again, have an idea of what you've reviewed so far. Any other track that you recall? Any other ones? So that is correct, but what is it actually called? That information is all sent up in this part of the spinal tract, spinal cord. What is it called? So this is the dorsal column. So all of the information is sent up here. It's somatosensory information, things from touch, sensory receptors. It's sent up the dorsal column medial lemniscus pathway, and that's going up. It's found in this dorsal region. It would be a funiculus that is found up here. We also talk about the spinal thalamic tract, and I'll get to that a little bit later on. It is also sort of a lateral projection. And then we talked about one other tract, spinal thalamic, and then what was the other one? More synapses. Yes. So spinal reticulothalamic, so it makes connections from the spinal cord up to the reticular formation in the brainstem, and from the brainstem up into the thalamic region, and that's an ascending pathway. So the three that we learned are the dorsal column medial lemniscus, spinal thalamic, as well as the spinal reticulothalamic, all sending different types of information along the spinal cord from the outside of the body through different sensors. This actually represents descending motor pathways. You should be familiar with some of these. They're sending information from the cortex down to the spinal cord, from the red nucleus down to the spinal cord, and then the reticular formation down to the spinal cord and the vestibular apparatus down to the spinal cord. In fact, we're going to start talking about these two down here in more detail starting right after your term test or midterm test on Tuesday. So we'll talk about these descending pathways. They're motor pathways. Every last descending pathway is a motor pathway, and these motor pathways are found in these regions of the spinal cord. So they're found, in other words, in the lateral region. So the corticospinal tract is a descending pathway that's found in the lateral funiculus, and these reticulospinal, vestibulospinal are found in this ventral funiculus down here on the bottom, okay? And this is a good slide for you to go back and do some review on. A couple of other things to review in terms of basic neuroanatomy that you want to make sure that you're okay with tomorrow. And again, you guys have 50 minutes to write your term test tomorrow. Is that right? So if you have 26 questions, likely you'll have some time to be able to review, but it's going to go by really quickly, in other words. So you should, again, go back, make sure that you understand the neuroanatomy of the funiculi, the ascending, descending paths in the spinal cord. Go back over the brainstem, which again contains the pons, medulla, as well as the midbrain. And sitting on top of the midbrain, what was the structure that we talked about? So if we go from spinal cord, and then from the spinal cord, we go into the pons, the medulla, pons, and then up into the midbrain. What sits on top of the midbrain? Yes. And the diencephalon can be further subdivided into the hypothalamus as well as the thalamus, right? So again, some basic concepts in terms of the neuroanatomy. Again, spend some time going over the cortex, a little bit about how we name different regions of the cortex and how we orient ourselves around the cortex in terms of the different sulci, the infoldings, in other words. But overall, it's really the spinal cord that you should be spending some time on. We didn't talk about too much of this year. So I'm going to ask you to make sure that you talk or are familiar with things like nociception as well as some of the touch, which I'm going to outline for you here. This is a good slide for you to make sure that you go back and review. So you should know what sorts of somatosensory information from mechanoreceptors are relayed into the brain through different types of axons, and we can identify axons by the size as well as the myelination pattern. Okay, and the exact numbers, like 50 meters per second or 10 or 1 meter per second, are not ones that you have to worry about memorizing. Again, mechanoreceptors versus thermoreceptors versus mechanoreceptors for pain, like the A delta C as well as C. The other thing I'm not sure why the slide didn't actually turn out, you should know the different receptor endings. So again, I'll mention Pacinian corpuscle, Ruffini endings, Meissner corpuscles. Wherever you see slides, you should expect like at least 1 or 2 questions related to the basic properties and the anatomy of Pacinian, Meissner's, Merkel discs, whatever was covered in your slides, you should make sure that you go over them. This is another slide that you should spend some time going over. You should know some of the basic properties associated with nociception. So in general, what do the nerve endings that signal for pain look like? Are they encapsulated? They're free, naked nerve endings. They have nothing except these proteins embedded within them, okay? And these proteins that are embedded within them might react to hydrogen, like ENaC. They might respond to molecules like capsaicin, as you see here with this TRPV1 molecule, or they might respond to acidity or hydrogen ions, and all of that is shown for you here. You should also be aware of the signaling mechanisms that would lead to them being activated. Okay, and I think I covered that in class for you, right? So make sure that this slide is something that you spend a little bit of time going through. Make sure that you understand that mechanically damaging tissue. I've hit my finger, I burned my hand, I've physically damaged the tissue. That physical damage in the tissue, whether it's in the form of hydrogen ions, whether it's in the form of heat, gets turned on and signals more because we activate this other GPCR that responds to ATP or bradykinin. It requires this PKC epsilon pathway that will further stimulate this TRPV1 that is found on those naked nerve endings. Okay, is that okay for everyone? Have you guys reviewed this? You know it really well? Kind of well? Not so sure? Okay, so again, you want to make sure that you know all the details on this slide. So for example, a good sort of way to think about this is as an integrative type of question, right? So if you're just thinking, how can you make this more complicated, or how can you ask the same concept on that slide but in an integrative fashion? Throughout this course, you guys have learned about different types of ions. Without looking at that slide, because I know you don't have these lecture notes in front of you, um, if I were to ask you, for example, if I were to activate the TRPV1, so you have to know what TRPV1 is. You may or may not remember it tomorrow, and this is just an example. What sorts of ions flow through there? So sodium is one of the ions that flow through. What are other ones? Calcium. So again, we've learned in other systems that sometimes things like calcium is really important in other signaling systems as well. Not in terms of exocytotic release, but actually for depolarization. Do you guys know any of the other places where we talked about calcium as potentially being important in the sensory system? So the bipolar cells are one place where we can certainly talk about calcium being important, and I'll bring that up later. So start thinking about it. If you know the signaling pathways, the sort of things that come in, you'll be fine. But those little details are going to be important for you as you will find out tomorrow. Again, you should make sure that you know the different axonal fiber types, whether they're myelinated or unmyelinated, whether they're large diameter, whether they signal for slow pain, if they're grouped together also in the spinal thalamic tract, or are they more likely to be grouped together, for example, in the spinal reticulothalamic tract. Should know and go back over the gate control theory of pain, as well as referred pain. And then I'll just give you sort of a broad idea that it's important for you tomorrow. That, as I mentioned in class, this is something that you probably want to go back and review a little bit of. This is another slide that you should make sure that you understand. So what system are we talking about here? It is not the somatosensory system. Sorry? So broadly speaking, this is the proprioceptive. It's about our joints and space. And you should know, again, from this section, the most important things, what is the receptor? So the receptor, as one of you have mentioned, is this muscle spindle. And this muscle spindle is the thing that actually gets activated by passive stretch. It's not you're actively stretching the muscle, it gets stretched for you. Someone either pulls your arm or a heavy weight gets put on your arm and it causes the muscle spindle and the muscle fiber to react. When that happens, if we were to measure from that muscle spindle that uses a really large diameter axon, do you remember, relatively speaking, is this a fast or slow type of response? It's very fast. So what do you expect this 1A fiber to be? The axon that's surrounding this muscle spindle, what type of axon do you expect it to be? Large diameter myelinated axon is the fastest type of axon that we have. It's one of the fastest reflexes that you have. And again, in your notes, you'll also find other things related to whether or not this is rapidly adapting. Those are things that you should also be aware of. Is just looking at the stretch response here, do you see that this is a rapidly adapting type of receptor? How many of you say yes, it's a rapidly adapting type of receptor? How many of you say no, it is not rapidly adapting? How many of you are not sure? So you pick choice E. Yes, there's no choice E though. Okay, so you can tell that there is a dynamic phase to it, so it's not consistent all the way through. So if it was consistent, this is what it would look like, but there is sort of a rapid dynamic phase to this, and then there's a consistent response, a tonic response to muscle stretch. So if it was just a rapid adaptation, just like in the thermoreceptors, when we have a stimulus, in this case, the stimulus is no stretch, suddenly there's a big stretch, that stretch continues on, and as soon as we take the stretch off, it stops responding. If it was a rapidly adapting type of receptor, we would see that during the initial application of the stretch or cold application or whatever it is, that it would respond early. Then if it rapidly adapted, we would see no response until we took the stimulus off, and then we might see a small burst of action potentials. Each of these up and down lines represents some electrical activity or action potentials that we can measure. So again, make sure that this is a system that you are familiar with tomorrow. So the last one, and this is a really busy looking slide, and it's busy for a number of reasons. So a couple of things to keep in mind. This year I did not concentrate on telling you about things like the anterior white commissure. So if I didn't really focus on that in lecture, you shouldn't really worry too much about that. This year, every year I kind of change up what I tend to emphasize. Should know a couple of things. For example, the spinal thalamic tract, the most important things that I've mentioned to you in lecture are what sorts of information does it convey? Always think about that. We talked about 3 systems. This is one of the 3 systems that I'm using as an example of how you should review. What type of information is conveyed along this tract? Does this tract switch, crossover, decussate in the spinal cord, or does it do it higher up in the level of, for example, the brainstem or the thalamus? Where does it do that? And then if it is traveling and sending information up the spinal cord, what region of the spinal cord is it doing so? So you can see that this is conveying information from the A delta axons and the C axons. So what does A delta and C mean? A delta is small diameter, myelinated, so they convey information relatively slow compared to the large diameter myelinated, but they're still 10 times faster than the unmyelinated small diameter C fibers. So these axons enter into the spinal cord through the dorsal horn as we'd expect. They will then send projections over to the opposite side. So this pain information is actually conveyed on the opposite side to where I felt it. If I hit my hand on the right, I would have release of noxious stimuli in the right. That information is conveyed over on the right-hand side, but then it crosses over. That same information from the pain information on my right crosses over to the left side of my spinal cord, and it goes all the way up my spinal cord until it reaches the thalamus. So again, what else? When you're looking at the 3 tracts, you only have to worry about 3. And in particular, the dorsal column medial lemniscus and the spinal thalamic, those are the 2 that you should spend most time on. What is the information? Where is it conveyed? And based on this diagram here, in terms of the funiculi, where is this information, this ascending information, where is it being conveyed? Yes. So this is more of the ventral funiculus. So what other in the ventral funiculus, what other types of tracts do we find, whether it's ascending or descending? We just covered it. So this is going up. There were other motor tracts coming down. Do you remember? Vestibulospinal was in this region, in the ventral funiculus. It was coming down, not going up, but it was also sending projections down here. And what was the other one? Yes. Yep, reticulospinal was also sending information down through this region. So knowing what area you're looking at is, again, also important. So this is primarily pain information, some information about temperature as well, going up through this region of the spinal cord. And the other thing that I asked you to make sure that you knew about was in the thalamus, different types of information gets conveyed into different parts of the thalamus. So we talked about the centromedian nucleus of the thalamus. We talk about the ventrocaudal nucleus of the thalamus, and we talk about others. Make sure that you know what information is getting relayed to specific nuclei up into the thalamus, okay? All right, so the last thing I'll talk about for the somatosensory system, I mentioned this again in passing. I only have 7 questions. There's a lot of material for 7 questions on this first group of 3 lectures for the first time that we met, right? So it was really kind of uh, trying to pick and choose and make sure that it was something relevant for you. Um, so we talk about this particular system. You should know, for example, how mechanical gating of the nociceptor response works. This spinal thalamic cell that you see here is this wide dynamic range neuron that we've also talked about. Without looking at this slide, and I'll just advance it one, the SG that was located there on that previous slide, what does SG stand for? And why is the SG important? So it's this substantia gelatinosa, and in this substantia gelatinosa, we have a number of these inhibitory neurons that we find here. So when you send a signal in, you're sending in a pain signal here, and this pain signal is telling this wide dynamic range neuron that is receiving multiple inputs from a number of different receptor types, that if I'm simultaneously activating mechanically. So I've damaged my thumb. My thumb is signaling that I have pain. And at the same time, if I'm mechanically stimulating, either rubbing or doing something else to the same thumb that I have actually damaged, that through this substantia gelatinosa, I will activate inhibitory receptors that will turn off some of that pain response. And this is that mechanical gating. That mechanical gating of pain actually involves this inhibitory system that you see here that relates to this substantia gelatinosa layer, or the SG layer that you see here as well. Okay. Do you guys all understand mechanical gating and how these inhibitory neurons can turn off a pain signal that's coming in through the C fibers, and you are inhibiting them through a large mechanoreceptor? Does that all make sense to you? So which track is the pain information coming from? There are only 2 up there, right? Um, the information from my thumb is coming in through which of those 2 fibers? If it's pain, it's the C fibers. C fibers, again, meaning that they are what type of axons? Small diameter, unmyelinated axons sending pain information in. So it's slow information coming in. And again, this mechanoreceptor that you see here is the A beta type. So think very carefully about this. Could that information have come from a Pacinian corpuscle? If you really know your material at this point, you should be able to answer that without any problems whatsoever. Pacinian corpuscle, yes, no. So this is a situation that you might encounter tomorrow, yes, no, and you guess. You're going to guess yes or you're going to guess no. You can't leave it blank because there's no penalty in guessing anyways. Is it no? It is no? How many of you would guess that this information from a mechanoreceptor is not coming in from a Pacinian corpuscle? It's not a Pacinian corpuscle. How many of you say, yes, it is a Pacinian corpuscle? Okay, so let's think a little bit about this, right? So we have cutaneous receptors on our skin. Most of these are somatosensory. They can signal through either the A alpha or the A beta. So do you remember the Pacinian corpuscle was signaling through the A alpha? So it's the one that conveys the fastest signals um, into the spinal cord, and the A beta types are the ones from everywhere else. So except for the Pacinian. So Meissner corpuscle, Ruffini endings, everything else from those mechanoreceptors is coming in through A beta. Okay, so That's somatosensation. And talking about the visual system, where you have another 7 questions totally independent of that. Again, a big section, a big block, a couple of things for you to make sure that you go back and make sure that you really understand. So you should know the different anatomical layers. Yeah, do you have a question? Yep, yep. Are they being sent up the intermediate zone? No, no. So a couple of things to make note of with the spinal reticulothalamic tract. One, I'm not going to be asking you the anatomy of the spinal reticulothalamic other than what kind of information might be conveyed. All of you should know that it's primarily pain information in the spinal reticulothalamic. Does it cross over? And in terms of the number of different uh, types of synapses, we find more synapses in that tract than we do in the spinal thalamic. But outside of that, this year I didn't really concentrate on it. Some years I spent a lot more time on it. This year we just didn't have the time to do that. Yeah. So again, you should make sure that you know the anatomical layers, the different types of accessory structures in the eye, different glands, how, for example, um, the different glands that are related to tear formation or to different types of fluid formation in the different chambers become important. You should know, for example, where you might find the different zonules, what chamber it might be located in. And again, you should make sure that you understand the anatomy of the eye that was presented to you in about the first 15 or 16 slides in the first hour that we had last Thursday. So make sure you spend the time going back and reviewing that. Make sure that you understand the different layer systems. How many layers in terms of tissue types do we have in the eye? So we have 3. Do you know all of them? Okay, perfect. And do you know what the major features or the major structures are that are associated with each of those layers? Yes? So again, when you're going back and reviewing, if you're thinking about this is sort of the inner layer, this is what I would expect to find in the inner layer versus this is what is found in the vascular layer, and this is what is found sort of in the outer fibrous layer, then you should be in good shape. But you need to make sure that you know what the overall structure anatomy of the 3 different layers are, the accessory glands that are associated with them, and where you might find different structures like the suspensory ligament. So it's anatomy / physiology. Should expect about 2 or 3 questions from that. Okay. Overall, I don't think that part will be too complicated for you, as long as you've spent time reviewing it, understand where all of these things fit in. So this leaves another big chunk of things that you should be familiar with. Again, I only have 7 questions. Whether or not they all get taken up, hopefully they do, but whether they all appear for you on your final or your midterm, I'm not really sure. So you should understand sort of the what's called the dark current, which we spent some time going through. Um, Dark current is okay for everyone? Yes? So again, you should make sure that you know in both the light and in the dark where you have activation of things like transducin, or where you don't have activation of transducin, where phosphodiesterase is really high or phosphodiesterase is not so high. Read through, and I'll be very honest about it. You need to really, really know this well in order for you to be able to answer the multiple choice question associated with it well. In other words, you need to understand what the role of cyclic GMP is in this situation. You need to understand the biochemistry, and you need to really know not just this slide, but other slides that are associated with it. Is this the complete story? No? What's missing on this diagram? Potassium channels, right? So, what other things are associated with depolarization, hyperpolarization, what other sorts of things are going on, and do you know like what would happen in a stepwise fashion? If you do, you'll find the question easy. If not, you'll have to spend some time thinking about it really carefully. So again, spend time going over this. There are some questions that are associated with it. So you must, in other words, know the current both in light as well as in dark. The cell types that are involved, the biochemical pathways for sure, and it's a must for this midterm. Is there a question at the back or no? Again, you should also know some of the concepts that are associated with the bipolar cells and the on-off bipolar cells that are associated with it. Again, do you expect to see a multiple choice question on and off bipolar cells? Why do you say so? Just have a good feeling about it? Um, I think that if you understand how these these rod cells, and it could be cones, how these rods and cones then interact with the bipolar cells, and how glutamate can either turn on or turn off these bipolar cells, and you know what an on bipolar cell is or what an off bipolar cell is, by definition, which one is which, in the presence of either light or in the darkness, you should be able to work your way through that question. Again, to be honest, the questions themselves are straightforward if you understand how photoreceptors, the rods and cones, respond in the dark, and what an on bipolar means and what an off bipolar means. Okay. And do you guys know which type of glutamate receptor is associated with the on and which is associated with the off? Can anyone tell me? On bipolar cells have what kind of glutamate receptor? mGluR6, and what's the other type? So it's the ionotropic. And again, you should be clear on that. On the slide, it actually tells you an on bipolar responds in this way and an off bipolar responds in another way. And you need to understand the receptor types and why it might respond in that particular way. Again, just to reiterate a couple of things. We didn't spend a lot of time this year talking about amacrine cells, nor did we talk about horizontal cells. Some years I spend more time talking about it. This year we really didn't spend a lot of time going through it. Is really just that outline slide that I gave you to make you aware of the different cell types, but you might want to just review that really quickly. This is an important slide for you. As I mentioned, you should make sure you understand this slide. So it's no use trying to memorize this slide. You're not going to be asked, which of the following is a characteristic of an on bipolar. You're going to be given a scenario, or as your colleague said, a case study. You're going to have a scenario where you're in the light or in the dark and something else is going on, and you should be able to work your way through the problem like that. So knowing what an on bipolar is important, but also understanding what sort of happens is also important as well. So it's a slide that you should be thinking about ahead of time. Yeah?
There was one slide where you talked about the receptive field.
Yep.
Like at the fovea, you said the ratio between ganglion.
It's 1 to 1. Yep.
1 to 1.
Yep.
But like, how does that work for the on center off surround ganglion cell?
There is no on off. It's literally it's just either on or off. There is no center surround. Nope. Yep. Okay, so again, this is your question about the ganglion cells in the fovea. So we don't really have that capacity to build these receptive fields to give you on-off responses. So again, know some of the details associated with photoreceptors in the fovea. This is another important slide for you to go back and review a little bit. Again, I didn't spend a lot of time on it this year, but it's something to be aware of. It might help you with some of the other questions that you see, but just in terms of how this responds, it's the retinal ganglion cell that we are measuring this response from, so that's important. It's not from the bipolar cell. The bipolar cells and photoreceptors help to make the ganglion cell respond in a certain way, and how this on-center, off-surround, when you stimulate across the entire receptive field of this one ganglion cell underneath all of this, that it's going to be weakly active for both scenarios, okay? And the last type that I'll mention, you should have some idea about the differences between M, P, as well as the last type of cells, which contain melanopsin, and you should have some ideas about how these different ganglion cells work and where they send their projections to, and how they sort of respond. Are you guys feeling more overwhelmed? I see a lot of, I need to go get some more coffee, I'll be staying up all night type of looks on your faces. Couple of other things to just wrap things up for today. One is, do you know how the different systems in the eye respond to either sympathetic or parasympathetic stimulation? Make sure that you review that. Someone was doing something that reminded me of one of the other things to, the reflexes in the eye. You should also make sure that you are comfortable with that. What reflexes did we talk about?
Pupillary reflex.
Sure. So the pupillary is also known as what else? Without checking your notes, because you should all know this by now. The consensual reflex, right? So what does that actually mean? Consensual in this case meaning if I shine a light into the right eye and I am testing for normal brain function, what would I expect in my right eye? If I shine light into it, what should the pupil do? It should expand as big as possible. It will constrict as small as possible. But that's only, the stimulation is only into the right eye. But what should I expect to see if I have a normal brain where nothing has happened? And again, you do this like if you hit your head, one of the first tests that they do is they shine light into one eye and then into the other, and they want to see both eyes. And when they shine light into the right, they want to see what happens to the left. And when they shine light into the left, they want to see what happens to the right. So what should I expect to see in this consensual reflex if everything is intact? Also constriction on the left. And why? More importantly, why? The midbrain is intact. Okay. How do I send signals to the midbrain? Okay, so again, make sure that you're completely okay with the anatomy that goes into things like these reflexes. And everything that you've heard about PSL and midterms and everything else, pretty true. So don't expect a cakewalk, but also don't expect it to be unfair. You won't see something from a slide that I've never covered, nor should you expect like a little tiny detail related to either numbers or something else that I told you to ignore to be on the midterm either. So if you can go through all the things that I highlighted for you today, you should be in at least decent shape for my section. I don't know about Dr. Key's section. He also tends to be pretty fair about it. It's all case-based, scenario-based application type. So don't expect just to see like list which of the following you don't see in the photoreceptor, for example. Okay. And if you can, try to have fun during your midterm, if you can. I know it'll be hard. Any other questions before we wrap things up? Yep.
Do you mind going over the cortical organization again?
So that's a big topic. It's like asking me to tell you vocabulary in
Just the fourth layer and the sixth layer.
So I got a lot of questions about that. I'm not going to be asking you like what's the output of like layer 6 or what's the output of like layer 4. You should know some of the details related to in the cortex. You have 6 different layers that are found in the gray matter. They send their axons out through the white matter to other regions of the brain. But outside of that, I don't think I asked you specific questions. If I did, I'll take them off, but I don't think I did. And it's outlined as per the slide. Yeah, if I did, and I honestly can't remember because I did it before. I think I actually gave that lecture. So yes.
Is all of area 3 considered part of the post-central gyrus? Or is it just 3A or?
So again, I'll consider it sort of all of 3 as being part of the post-central gyrus. Yep. Any other questions? And area 3 being important for what?
Oh, it's for the 3A is for cutaneous receptors. 3B, no, sorry, 3A is for proprioceptors. 3B.
Oh, so another thing to keep in mind that you should know about different association areas. So outside of the main areas, so you have things going into specific nuclei in the thalamus. Know all of those. You should know where the targets are in the different parts of the postcentral gyrus, the different areas like area 3A, and then some of the association areas and what their primary functions are. Okay. Anything else? Yes.
In the first lecture, you talked about medullary shifts, and I was just wondering why that happened and what really was important from that slide.
It's really just to give you an overview of how again, you can look at it really simply. The spinal cord is a continuation of the brain, and this brain structure that we have, the way that it's organized is you have this central gray region here, and then we have white matter surrounding it. In the brain, if you were to take a look at the cortex, the 6 different layers are arranged as the gray on the outside. They send their axons here, and so we have gray matter and then white matter. So in some ways it got flipped around. Part of that process, that transition from being central gray and outer white to being outer gray and inner white, occurs at the level of the different at the level of the medulla. So the next part up from the spinal cord is sort of the brainstem. And as part of the brainstem medulla, some of these nuclei start to shift, and by the time we get all the way out to the brain, it's shifted completely. The organization we see here is different than up here, and again, it was just to give you an overview. Any other questions? So if it was a full-blown like a neuroanatomy course, I would ask you to know, for example, where the nucleus tractus solitarius is and what regions it is, but it's not. It's more of a physiology course, so you don't have to worry about it. Any other questions? Now's the time to ask. So if you were to give me a number between 0 being I will go home today and start studying versus 100%, like I could write the exam right now, how prepared are you? Well, this is a really quiet group. You're 50, 60, 60, 60. 60 seems to be the number. Okay. How about by this time tomorrow? 100%? Yeah? Have you at least gone through all the notes once? Okay. Anything from my notes that you want some further explanation on? No? I will be around in the office until about this time tomorrow. So if you have any burning questions, it's better to drop by, to be completely honest, than to send me an email, because sometimes I have no idea what you're actually talking about. Like, I think now in retrospect, I understand what the question was about, do we need to know all 3 nuclei in the thalamus? And I think it was related to this particular slide that I probably stuck up there. It was related to this, like why do you have like 3 different colors? Do we need to know all 3 up here? But it's really, I didn't really understand the question until I saw this slide right now. So if you have any questions tomorrow up until about 3 or 4 o'clock, drop by the office. I'll be happy to try to explain things. If you have any burning issues, I'm usually there from about 10 to about 3. So hoping that you have maybe 1 or 2 small brief questions to clarify things. I won't have to be reteaching you things. If I have to, I will, but I'm hoping that that last minute into the brain thing doesn't work well. Okay, don't know what else to tell you. Good luck tomorrow.